So we're talking about microbes versus the refrigerator, right? And so why are we even asking this question? And the answer for that is because we are talking about food. The refrigerator was invented to help people keep their food fresh. And so the modern refrigerator was invented in the 1830s. And it didn't make it into kitchens commonly until about the 1920s. But after the 1920s, a refrigerator was commonplace in people's kitchens all over the world. And the way that a refrigerator works is that it keeps food at a low temperature. Usually that temperature is around 4 degrees Celsius. And that is supposed to keep foods fresher for longer periods of time. Well, why does that work that way? Well, keeping food cold actually keeps microbes from growing very fast. So if you have something in the refrigerator, the microbes will grow a lot slower in the refrigerator than they will grow if something was left at room temperature. So in essence, you have fresh foods in the fridge versus moldy or you might say that this is spoiled food that would exist at room temperature if you don't eat it on time. And so the benefits of the modern refrigerator is that it keeps the food cold, which keeps the microbe growth low, which keeps food fresher for longer periods of time. But if you look at the human history timeline, we know that Homo sapiens, which is the species of us human beings, we emerged across the evolutionary timeline or the geologic scale about 200,000 years ago, right? And that's where we have it here. But the invention of the modern refrigerator took place only 100 years ago. So what was taking place in all of this time where humans had to have food in order to survive, but how did they keep food from going bad? And so what did people do when the refrigerator did not yet exist? And that's the question that I want to really uncover here. So let's introduce the small but mighty microbe to answer this question. So we know that a microbe is a living organism, and typically any living organism that is too small to see with the naked eye is considered a microbe. A microbe can be unicellular, meaning it has one cell, such as these bacteria here or these bacteria here, or it can be multicellular, where you have several cells sort of living together in concert and they're functioning together to make sure all the homeostasis and the metabolism is going well. Now, the cool thing is that microbes are actually found everywhere. We know that they are in the dirt, they're on the table, I can get them from the outside of my glass. They're in water, they're all over our bodies. In fact, our bodies have about 2 times more microbial cells than human cells. That's how many microbes are in our bodies. And when I say the built environment, that's what I'm talking about like, floors, carpets, walls, things like that. And so how do we think about keeping food fresh and nutritious by leveraging all of the microbes that naturally exist on the surface of food, right? And that is basically how people had thought about keeping food fresh in the olden days, although they may not have really considered this. They weren't like, well, we're gonna keep food fresh by having healthy bacteria grow and outcompete the bad bacteria or the bad yeast. That people weren't thinking about it that way, but essentially, we can use the natural microbial makeup that already exists with foods to help keep foods edible and nutritious and even make great flavors. And so what do I mean by that? Well, as I had mentioned, if we bring back our little timeline, we know that the evolution of Homo sapiens took place around 200,000 years ago. And we know that the modern fridge was introduced about 100 years ago, right? And so a lot of people who study a topic called anthropology, they were trying to figure out, well, what evidence of food preservation might we find from the historical records? And the oldest example of food preservation was actually found in ancient China in 7000 BC, and it dates back to about 7000 BC. And this is because if you think about human civilizations, 9,000 years ago, 10,000 years ago, think about what they did not have that we have. We have electricity, we have plumbing, we have places to wash our hands. We have waste that's separated from our food and drinking water. And so, it was really important. And for us as modern humans, we have the capacity to make sure that all of the things that need to be healthy, like our water, our food, is clean and not being contaminated by other kinds of like more pathogenic or more unhealthy kinds of microbes that can make us sick. So 10,000 years ago, they didn't have any of this stuff. They didn't have electricity. They didn't necessarily have modern plumbing, and they didn't necessarily have a modern understanding of how diseases even work, right? And so, but they did know that if they went and drank some water from, a local watering hole, that maybe the water would make them feel sick. So what they did instead, a lot of time, is they fermented things using the natural microbial makeup of the food that they were fermenting. And that helped to basically kill anything that made you sick and also raise the amount of microbes that can actually be symbiotic for your body. And so in ancient China, about in 7000 BC, they made this beverage called kui, which is made from rice and honey. So when you ferment, you need to have a sugar source, and rice has a carbohydrate, honey has a lot of sugar associated with it, and the fruits of grape and hawthorn plants. We see this again even for a little bit later in ancient Babylonia, which is now known around Western Iran, where we found the first recipe for beer. And beer is a fermented beverage. And again, beer was brewed to drink and be able to sort of hydrate yourself because drinking water was not necessarily safe. A cool fact about ancient Babylonia is that the people who brewed the beer could only be women. And they were revered as being a brewmistress was like being a lawyer or a doctor today. It was a very well respected profession. And then the Egyptians, we see not only were they fermenting beverages such as beer, but they started also fermenting things like flour to make bread. And also they fermented a bunch of dairy products, making things like yogurt and cheese and butter. And so when you're fermenting these things, what you're doing is you're taking milk, for example, and you're adding a bunch of really good microbes that are helping to preserve that food so that the milk lasts longer and has more nutritional value over time. So we've seen culinary history of fermented foods across human history, from the ancient Babylonians to we see vegetables getting fermented in China. Um, you might have heard of kombucha in the grocery store or bodega. There's plenty of that going on now. And we also see examples of products like miso and dosa and sake start turning up around 500 to 1000 AD. And so why go on about all of this? Well, if you think about how we eat, and especially if you were baking bread in the pandemic like I was, you started to become pretty intimately familiar with microbial communities. And microbes are so important, not just in food, but they're important in keeping our bodies healthy, and keeping our environments healthy. And so I think it's really important that we talk about them. And the most exciting microbe that I love to talk about actually is one called yeast, right? Yeast is a eukaryotic cell, which means that it is in the same kind of categorization as human cells. An example of a non-eukaryotic cell would be a bacterium, which would be a prokaryotic cell. And we can go into that difference in a little bit. But essentially what yeast does is it uses the flour as a form of energy, and it creates products such as ethanol and carbon dioxide. And it's this carbon dioxide that you can see all the bubbles in this starter culture that contains yeast and flour and water. And these were used to bake bread. And all of those bubbles are actually represented in, if you slice a piece of bread, all of these sort of holes that you see in your bread are because of the carbon dioxide that the yeast were expelling from their biology and their metabolism. So if you were going to take a ferment and look at it up close, and this is an example of what you might find in a sourdough or maybe a kombucha, where you see a bunch of little tiny organisms just sort of hanging out. And you can tell a yeast cell because it's round and globular, and you can tell a bacterial cell because it's elongated and it's a little bit, so these are all sort of these little stringy things. They look like little sprinkles. Those are all yeast cells. Sorry, those are all bacterial cells. And they, together with yeast cells, they live in this, and it's called symbiosis, where they sort of help each other and they produce materials that other organisms can use for fuel and so on and so forth. So it becomes this really beautiful interaction. But what I want to do now is go into some of the really cool imagery that we have on Snorkle to take you through this a little bit more fully, right? So here we have our
Right, Jean, I just
yes,
want to make one interjection here, which is that
sure.
you can actually scan the QRs on the bottom right with your phone to have the same interactive visuals that Dr. Garbarino is going through. So,
Yeah. I hope you'll join me on this. All right, so Uh, here you can see the yeast cells. Let's go into a yeast cell. Let's look at that. And so I love yeast cells. I actually studied them for a very long time. I used yeast actually to learn about the human body as it relates to diseases like diabetes and heart disease. And that's because yeast are, they have a conserved mechanism in that a lot of the genes and a lot of the processes that takes place in yeast are actually almost the same as the processes that take place in a human cell. So you can use them to learn about human biology as well. So this is what a yeast cell looks like, right? So this is your typical very healthy Saccharomyces cerevisiae. You can see that it's pretty round, right? It's covered by actually a cell wall. And then it also has a cell membrane. It's kind of like how plants have cell walls? Well, yeast actually also have cell walls. And so inside of the cell membrane, we see a couple of these cool things. They're called organelles. Yeast have this blue thing here is a very large organelle called a vacuole. Whoops, zoomed in a little bit too much there. And the vacuole is sort of like its garbage disposal. It helps to take the things that, it plays a really large role in keeping the garbage sort of recycled, and all of that material gets put back into the yeast cell so the yeast itself can use those materials. Yeast also have things like mitochondria, which are the powerhouses of the cell. Yeast have an endoplasmic reticulum. You might have heard of that in school, a nucleus, a Golgi, all of the things that you would expect to find in an animal cell, a yeast also has. And so what's happening here, just so we can talk about the shape here for a second. So the yeast, they reproduce by budding, right? And so this would be a mother cell that is in the process of budding off a daughter cell. So they don't divide equally, like two halves just sort of divide, and it's sort of an asymmetrical division. Okay, so let's see what that actually looks like. So you can see here, this is a yeast cell that's about to divide. Maybe this is a yeast cell that doesn't have a bud yet. And if we were to sort of just like scroll through that, you can see also, you see these dark places here. That's actually the vacuole. It's one of the largest features of a yeast cell. So you can see here that the bud is coming on a different plane from this cell here. And you can see how they grow. They sort of just blip is the technical term. To be honest with you, that's what we call it in the lab. The daughter cells blip off, become their own cell, and become mother cells themselves, and then blip off again. So this is called bright field microscopy, and it's one of the things that we use to really just understand the features of cells just by looking at it. But another cool thing, and so as you can see, just to back up, you can see how those 2 or 3 cells becomes a huge clump of cells in just a short amount of time. So one yeast cell can become 2 yeast cells in 90 minutes. So if you were to double that and double that and double that over a 24-hour period of time, you'd end up from one cell to many cells. And that's one of the powers of microbial growth. All right, so this is an even cooler visual. So this is that same bright field, except the yeast actually were dyed with different kinds of fluorescent probes. So the green is dyeing the cytoplasm. The blue looks like it's dyeing the vacuole. And the red is dyeing the genetic material or the nucleus, where the nucleus is found. And so here's another example of the cells dividing. And one of the things that I want you to look for as I scroll through this is the red. You can see how the genetic material, yeast have DNA, they have linear DNA. You can see how that DNA gets transferred into the daughter cell and it's really, really cool. So you can kind of see it right here. So it's transferring from this mother cell to the daughter cell as one example. You can sort of start seeing it taking place here, but I'll keep on scrolling and you can sort of see how cool it looks. I think this is, if I had access to Snorkle as a graduate student, I may not have gotten any of my work done because I probably would have been playing with this the whole time. But I love the way that it sort of just flies in. But let me move it a little faster so you can see it. I don't know if you saw this, if you pay attention right here, this one's a really beautiful one. I love the way the daughter cell blips off right there. So anyway, you can see what that looks like. And you can see how the genetic material goes from you can really see it shooting out here. The genetic material or DNA goes from the mother cell to the daughter cell, it blips off and then becomes its own organism. And these are in fact clones of one another. There is no mating taking place here. One of the cool things about yeast is that they can exist very healthily with one set of chromosomes or 2 sets of chromosomes. The benefit of having one set of chromosomes, especially as a geneticist in the lab, is that I only need to manipulate one set of chromosomes and not 2. So it's very useful in the laboratory. And then the last thing I wanted to show you, so we talked about the yeast budding, and we talked about how the genetic material goes in from the daughter cell to the mother cell. But I want to show you one of the most beautiful things that happens. And I think every mother out there might be able to relate to this, and that is the scars of your children. So this is a really cool electron micrograph of a yeast cell. And each one of these cool little circles is actually, oh, I'm very bad at the Zoom, I'm sorry. Each one of these cool little circles is actually a scar where the daughter cell budded off previously. So this yeast cell has divided 1, 2, 3, 4, 5, 6, 7, 8 times. So it's created 8 different daughter cells. And I know that because of these cool features that almost look like craters of the cell. But the cool thing about this microscopy, and so this really is a much, much more resolved image than the bright field that I showed you previously. You can see all of the details of the cell wall and how the bud scars have this sort of like folding over around this crater-like feature. And so, yeah, yeast are incredible. They help us to learn so much about people, and they help our foods remain preserved, and they help our foods to keep from being spoiled. To be honest with you, one of the coolest things about yeast is that they prevent if they're in symbiosis, like in a cheese or in a yogurt or something like that, they're the ones that are helping the good bacteria also work together to create those rich and healthy flavor profiles. So it's very, very cool. All right, so that is the end of what I have to share, and I'm more than happy to take any questions that you might have. And thank you for your time tonight.
Wow. Well, a virtual round of applause for our speaker.
Thank you.
Thank you,
Sorry
Dr.
about the sharing of the screen. I can't believe that. I should be a Zoom pro by now, but
Oh, that was amazing. Yeah, so we can open it up to some questions if you have.
is there, oh, I love this. Is there a limit to how many times a single yeast cell can blip? And I don't actually know the answer to that question. I think that there probably is eventually some sort of limit. Um, but I think it depends on how much food is available, what the environmental conditions are like, and how the other sensory kinds of elements in that yeast are playing out. You know, yeast age just like we age, and so there's probably a metabolic cutoff for division, but it's something that I can look into, and I can get back to you on that actually. So that's a great question. Thank you.
Great. Other questions?
One of the coolest things I know I see there's another question here, but one of the coolest things that I do is I cultivate wild yeast from the environment. And so the way I do that is, oh, well, I don't know if you've ever been to a grocery store and you've gotten a bag of, especially like purple grapes, because it's easier to see. You can see how there's that sort of white sheen on top of those grapes. Well, that's actually a yeast bloom. So that is one of the easiest sources of yeast to find. And all you would need to do is take that one single grape and you can put it into a vat or a little cup of orange, sorry, not orange juice, apple juice. And apple juice is actually the perfect medium for growing yeast. And during the pandemic, this is how I taught all of my microbial genetics courses, is I had my students who couldn't be in person go outside and collect things like leaves, um, grass, different kinds of dirt, different kinds of fruit, maybe some dog hair, I don't know. Put it in some apple juice because apple juice will help select and enrich a yeast population. And we made starter cultures and then did a bunch of genetics off of that. And so, you know, yeast, the natural source of yeast is actually an oak tree. That is one of the first places that yeast symbiosis is thought to have occurred in sort of the modern history. And so even if you went and got an oak leaf or some oak bark and popped it into some apple juice, you'd have a beautiful yeasty situation. Okay, so there's another question. Since you bought a beer, is there a biological reason behind why beer gets spoiled by taking it in and out of the fridge too much? I don't know the answer to that question either. Shirley, you're asking very hard questions. But I would call, I mean, I think I know that the common like colloquialism for that is that your beer gets skunked. Because it's apparently if it goes in and out and in and out, it like, I don't know, it has some sort of, it must be some sort of microbial imbalance because it has that smell to it. And it's usually the microbes that are making smelly things. So, but that also a great question.
Okay, do we have other questions? Well, if not, let's thank our speaker again.
Oh, there's one more question, Jonathan. Should I answer
Questions?
it?
Oh, yes, go for it.
What makes the process smelly? This is the best question. Okay, actually the process of fermentation. I love this question, Shirley, because when yeast are, or any microbe like lactobacilli, which is the very popular microbe that you might find in like your yogurt in your refrigerator. So anytime they are creating energy, so they use sugar to eat, right? And the sugar then goes through these metabolic processes, and ultimately you want the sugar to get converted through a stepwise process into a molecule called ATP. And ATP is the energy currency of almost all life, if not all life. And what happens during that process of going from sugar to ATP is that the microbes are generating waste products, actually. All right, so lactic acid, which is what makes your yogurt all tangy, is a waste product of converting sugar to ATP in the bacterium Lactobacilli. Similarly, when yeast eat glucose, so you go sugar and they want to go all the way to ATP, a byproduct of that is both ethanol and carbon dioxide, right? And so anything that's smelly that's coming out of these bacterial or microbial species is a waste product of the fermentation process. And so we have different kinds of cheeses, for example, that smell like baby vomit, but are apparently like very high-end cheeses. That baby vomit smell is the result of the microbial fermentation and the waste product generation, for example. So Shirley, I can answer that question. What makes the process smelly? It's microbial waste that we love to eat because it tastes good for us. Okay, when we're talking about wine, are we talking about the smelliness of the wine? Because there's a couple of things there's a cool story that I can share with you about winemaking. And essentially, if you are winemaking, right, you want to have a yeast strain specifically because you want to create the ethanol for it, but you also don't want to have the carbon dioxide get trapped because not all wine is carbonated, right? And the way wine sours is one of the ways that wine sours is that you have an uncontrolled growth of other kinds of microbes, such as lactobacilli, which create lactic acid. And so if you've ever tasted your bottle of wine and it tastes like vinegar, that's because the yeast have sort of the yeast that were there and created the ethanol, they've limited the amount of energy that they can create and they start sort of dying off. And once those yeasts start dying off, the lactobacilli get to there's nothing in their way. So they get to start growing. And it could be lactobacilli, it could be acetobacter, or other kinds of things that make these like smelly, vinegary kinds of products. Like acetobacter makes acetic acid is vinegar, for example. And so it's actually all about microbial competition. And Margaret asks a question on if there are many yeasts. Actually, there are numerous species of yeast. We work with the baker's yeast in the laboratory called Saccharomyces cerevisiae, but there's natural yeast all over the place. There's my favorite name is Zygosaccharomyces cerevisiae, actually. And that is the wild yeast that you might find in and around and on us. But there's a Schizomyces pombe, for example. There's Candida. And if ladies out there know what Candida is, it's a pretty nasty yeast, and it often can, if you've had a baby and they have thrush in their mouth, women can get it for yeast infections, things like that. So there's pathogenic yeast, there are symbiotic yeast, there's yeast that mean nothing to us, but there are definitely yeast, many, many different kinds of species of yeast all over the place. So, and across evolution, actually. There's extinct species of yeast, there's a species of yeast that haven't even evolved yet. So it's a very diverse segment of life. But thank you for that question.
I don't know if you can hear me.
We can.
You started out talking about how microbes can destroy food or make food unhealthy.
Yeah.
That's why we have refrigeration. Is there some way that you differentiate between healthy and unhealthy microbes?
So, yeah. Sorry, thank you for your question. All right, is there any other piece of it, or are you good?
That's it.
Great. So it's all relative, right? It's what makes us sick and what doesn't make us sick. But also what makes us sick may not make like your dog sick or your cat sick, right? So every species is susceptible to a different array of microbial communities, right? And so there are tons and tons of microbes out there, and most of them have no impact on our bodies, right? And those are called just sort of they're called symbionts, but they're neutral, right? They don't have a plus or a minus, right? Then you have mutualistic symbionts, things that we give, like our bodies provide some shelter, some resource, but their metabolism provides us with some nutrient or protection, as an example. Like you've heard of like healthy gut health, something.
Yeah.
But then there's the pathogenic stuff, And that's when either it becomes pathogenic when it becomes too many in our body. Like there's always candida in our bodies. It's when it becomes out of balance that it becomes, problematic for us, for example. And so it's all about what makes our system sick. And so E. coli, right? There's E. coli all over the place, but there's certain strains of E. coli that are virulent, that are not virulent, but they make us a lot more sick than, your typical E. coli that you might use in a laboratory setting. So it really just depends on how our immune systems are structured and what kinds of presentations those microbes give to our immune systems, and then what we do with that, right? And so, you can probably walk into but for me, I have when my kids were little, they would get all these colds from being in daycare, but I wouldn't get sick because these are things I had already been exposed to. So there's also like an exposure element of it as well. So I hope I answered your question, and I'm happy to elaborate if you have a further question.
Thank you. It's very helpful.
Yeah,
Thank
my pleasure.